a really broad subset of the academy. So Dr. Duncan Irschick, who's visiting from UMass Amherst. And so Duncan actually grew up in San Francisco and spent a lot of time as a kid coming to the old academy, and this is his first time seeing the new building. So hopefully he'll have some time this afternoon to explore a little. And this morning we were working with some folks in the aquarium to use one of the arrays that Duncan's going to talk about today to take really nice images of some of our reed frogs, which I've been working on. So it's been a lot of fun getting to know Irschick today, and I'm excited for you all to hear from him. So like I said, he grew up here in the Bay Area, and he did his undergrad at UC Davis and then moved to Wash University to do a PhD with Jonathan Losos, and has been at Amherst for a while now. And so he's definitely a herpetologist by training, but this new interest in really leading the field and developing these new tools and techniques for imaging organisms in 3D and living organisms in particular has been a growing interest in his lab. And so that's brought him into studying all kinds of animals. But hopefully we'll be pulling him back to the dark side of herpetology with our very cool reed frogs. So thank you so much, Duncan, for talking to us today. And he'll be around this afternoon as well if you'd like to follow up with him and ask more questions about his work. So thank you.
Great. Thank you, Raina. And it was lovely to be pulled back to the dark side, because whenever I work with reptiles or amphibians, that positive energy comes back. And yeah, I do have colleagues who, I won't say they've disowned me, but they've definitely wondered, why are you working these other animals? And I'll explain why. So I just want to thank Raina Bell for setting this up. And it's been lovely to work with Raina and her group. Megan was awesome in helping out with the frogs and the staff. So everything I can see so far is everything is very first class, an amazing experience. Congratulations on your renovations. I did grow up coming here. I used to go to the giant, I don't know if you want to call it the giant pit. I used to call it when I was a teenager and watch the giant alligator snapper, and that inspired me. I loved it. It's one of the main reasons, honestly, I became a herpetologist. I just loved walking around. Can I see the king cobra? Can I not? I think these experiences matter to a young person. They matter to me. I love these animals just as much today as I did back then. So in that light, the passion for biology, the passion for diversity is really what inspired this most recent project. And that is about the process of creating lifelike 3D animal models using scanning and software technology. And I do want to emphasize, it's not just a hardware approach, it's a software. It's also ultimately, I believe, a conceptual approach. It's one, I think it's also very highly collaborative and draws from many lines of inquiry across art and history and culture. I've certainly been influenced by those, as you have, I'm sure. The key aspects are implications for conservation, natural history, collections, and locomotor studies. So I'll talk about all these elements in turn. I certainly do want to emphasize that none of this work would have been possible without an amazing group of people. Undergraduates have played an absolute critical role. Some of them have been co-inventors on patents and technology, which was a thrill to me. I love to see when young people do exciting things. But collaborators, my African guide up there, I think Wilson was his name, got turned upside down, but he was the person who helped me go find and work with elephants, for example. Mark Mandika for the Amphibian Foundation, Cameron Seiler, so many other people along the way. And I think, to me, this was the most fun part of the project. It's like I talked about with my own kids when I work as a coach. I say, hopefully you're not just being a teammate and being a friend. And I definitely feel the same. So the friendships were critical to me. So a concept that you're going to be very intimately familiar with here at this Steinhardt Aquarium and California Academy of Sciences is digital heritage. I was just walking by with Raina and we seeing the photography of herbarium specimens and I smiled. And I think this concept means many different things to many different people. But I'll just give a few examples, some of which you may relate to. Shown in the upper left is a 3D laser scan of downtown historic parts of Philadelphia. There's a lot of 3D scanning and digital heritage of human artifacts and human landscapes, because some are very impermeant. For those, we're talking about politics, how you can't talk about politics anymore, but if you miss Obama, don't worry, you can get a 3D scan of him, which Autodesk did. Actually, they only scan the front part of his head. There it is. And you can download it and stare at a bomb in 3D. And so there's many, digital heritage is not just 3D work. It's really about, I would argue, taking things that are ephemeral in our human world, whether they're living animals or they're artifacts, and creating some kind of digital copy. And one of the best examples I can think of are old Hollywood movies, which were made on film. They were highly flammable, and all it would take is a match and the whole thing would just immolate. And so there's a lot of work. You can watch on Netflix some of the old digital archives. And obviously here at the museum, Stern Aquarium, you guys are doing an amazing amount of work in digital heritage, and keep up the great work. What I'm going to be talking about is specifically 3D scanning as a form of digital heritage. And 3D scanning doesn't just happen in one way. Some of these, you've probably been to the doctor's office, you've had it done on you, and you didn't even know about it because they don't really so readily share with you these data. You know, when you get a CT scan or MRI, they just say, well, you're fine, you're not going to die. But the reality is what can be created with these technologies, such as, and some of them you are going to be more familiar with than others, such as CT scanning or MRI scanning, these incredible 3D models. One of the whole motivations behind our work was we saw all these technologies out there. We noticed how they weren't really being used as widely. The techniques, the knowledge, and the expertise were not being shared as widely. There's also a mindset about the way these things are used. There's a concept about the way you CT scan something and how you use it. And we wanted to sort of change that. Other examples are laser and white light scanning. Here's an example down below of a laser scan from, I think, a historic building, I believe it is. And then some 3D photogrammetry, which is what I did with Raina this morning. That's where you use camera technology to convert essentially a 2D image to a 3D model that has both shape and color. And I actually took the time to go to Sketchfab and download some of the beautiful 3D scans that were created here at the California Academy of Sciences, which you can see down below. So the reason I've become very interested in 3D scanning is my basic interest is shape. That's pretty much what I've spent my whole career studying. I started out working with lizards and reptiles and ultimately taking a pair of calipers and measuring them. But I became very interested in new ways of thinking about shape. And that, in this sense, we can really start to think about some of the advantages of 3D scans. They provide a 3D shape map of an organism, or at least they have that potential to, at least the external shape. The very new part that I'm going to talk about today is they also now potentially provide a color map of an organism. And more importantly, the two can be joined. So you have a shape map and a color map that are linked into one another. And this is a new innovation, and you think about it, because a photograph, in a sense, is a 2D color map, but it's a 2D color map. And if we can take a 3D scan and then reconstruct its color, this would be an exciting innovation. Obviously, with people who've done 3D photogrammetry as a modeling process, they've known about color for a long time, but it's always been almost an afterthought. Like, oh, it looks really cool. It looks lifelike. It's awesome. But the emphasis has always been on shape and look. It looks awesome. But what about using that color as a scientific tool? One of the things we started to do was taking what basically static objects, like you take a CT scan and then you make it move. We had colleagues who studied them, thought that was very disturbing. But the idea is that you can be modified to add movement, and we can now study movement in the field. Can we add to them? And the final result, as we'll show you, can be strikingly realistic, and this leads to possibilities of creating digital avatars of animals we work with in the field that can have all kinds of value to museums and otherwise. And they can be 3D printed, which is great for education programs. We have a program where we have kids that do 3D printing and painting with. But what about living animals? If you were to go into the field, capture a frog, or you see an elephant or a shark, could you scan them? Could you create a 3D reconstruction of what they looked like and what they moved like? And in some senses, you guys are already doing this in a way. You're taking that steps. This is, again, a 3D scan here for the California Academy. Here's a scan of a dead frog from the University of Florida. At the bottom is a beautiful 3D scan of a Komodo dragon from the Natural History Vienna Museum. It's not living, but you wouldn't know it from the scan. It's a lifelike model. But how do we take that next step there? And what I'm going to be talking about is really a focus on external shape and form and color for a living animal as you might see it. And this raises the question about when you work with animals like a sea turtle or a marine mammal or a shark, these animals tend not to be well represented in museums, certainly not as whole specimens. Some of the specimens are here, they're often quite old, they're often quite warped. In the case of whales, for example, there will be museum specimens, they tend to be bones. But could you, for example, create 3D models of them and use them for science and conservation education? But even for animals that are more common, like this toad down below, those can be preserved, but well, obviously when you stick an animal in formaldehyde, they're going to change. Color is going to change, physical attributes are going to change. So this raises the question, what could you do if you had exact animal avatars? And hopefully the question answer, I would love to hear from all of you. And there's many different points of view on this. There's not just one point of view. This is a rhino that we work with named Bakari. Bakari is a male rhino from the Perth Zoo. This is one of the sweetest animals I've ever worked with. After we worked with it, they said, do you want to pet Bakari? I was like, can I? And it was just cool. But southern white rhinos, obviously a very endangered species, as all rhinos are. We were able to create a 3D scan of it. I'll talk about that later. But let me give you an example of a 3D scan that we did with a very critically endangered species, which is a leatherback sea turtle. This is an adult female. Her name is Mac. She was found nesting on a beach in southern Florida near Juneau. We were able to go out with camera systems, and turns out she was a very rare turtle. She was actually nesting during the day. The partners that we were working with were very interested in a 3D model of Mac because they said, if we could create a 3D model of this very rare animal, we can study tagging, we can study locomotion, we can study potentially body condition. These are not animals you see in aquaria. They're also not animals that you can collect. You can't even really hardly touch them. You come in, they nest, and they leave. And you don't see them in the open water either very much. So they're very challenging to work with. What we can see with this animal is what we're able to reconstruct for this particular specimen is the accurate shape, and we can prove that with data. I'm going to talk later about color and motion. For this particular specimen, the color is very accurate. It's depicted in the swimming in this ocean environment. I'm going to give some more examples later. So this kind of, that model, by the way, is openly accessible to anyone to download and is currently being used for a lot of different scientific projects. But let's say we could do these. What could you do? Well, I'm going to throw some ideas out. You could collect 3D models of organisms in the field. You could maybe swab their DNA and let those animals go. That could be a possibility in some areas where they don't allow you to take specimens back. You could perform computational fluid dynamics on 3D models of flying and swimming animals. You could rig animals, use them to understand motion. You could, and this is probably the biggest interest in our work. I'll talk later on about rhinos. We're also doing some work with elephants, is body condition. How do you assess whether animals are gaining and losing weight? And you could obviously use them to study new kinds of robots, and they'd be great for education, which we've seen already. So the goals of this project that we did, and this is called the Digital Life Project. It is an open access project. It's designed to recreate the shapes, colors, and motion of living animals. This is a very ambitious goal that's probably won't be achieved in my lifetime, and it's also a collaborative goal. And then to provide these 3D models to scientists and the public as open access tools. Now, all scientific data technically is open access. One thing I've learned is that's not always true because you go into these open access. There's a big difference between making something open access and making something very easy for people to use. And our goal is just to make it easy. And that turns out that has been a great benefit to others. And finally, to develop the methods for recreating living animals and to share these widely. I was talking with Raina about the possibility of working with her in a group, and it's not just us doing it. We also want to share it. That is obviously a critical part of what we're interested in. We want to train others, help others, and learn from them as well. So 4 main parts of this talk. So I'm going to talk about the methods. I'm going to talk about shape, and then motion and color. Of these 3 elements, shape, motion, color, really shape is by far the most developed, but color is becoming very well developed as we speak, and motion is still a work in progress. So to give a little background on 3D photogrammetry, most of you maybe have some familiarity with it. And it's not the only technique that we use, but it's the one I'm going to talk a lot about today. It's essentially a technique, this was from Zoo Atlanta, some work with Joe Mendelson. We had a poison dart frog, Phyllobates terribilis. You have a rig, you take multiple photos of this particular individual. He has tadpoles in his back. It converts those 2D images to a 3D mesh and then adds that color back on, like a 3D map, and you get this strikingly realistic 3D shape and color of the animal. Obviously, whatever cannot be photographed cannot be reconstructed. For example, the nether regions of the frog are not going to be reconstructed. We actually have now developed techniques to reconstruct those regions that are not seen here using other methods. So this really requires hardware. It requires cameras or videos. In the case of obviously field work, we want to be able to take this technology to the field, to the tropics, underwater, if we're going to work with certain kinds of animals. With certain kinds of animals, like what we did today with frogs, obviously you can bring a gear to a place like an aquarium. But this was actually a part where undergrads played an absolutely critical role. Up in the upper left, we have Dylan Briggs and Casey Smart, two undergrads who first developed a hand-built system. That was what we originally called the Beast Cam technology. And that was a 4-camera system that we could walk around and photograph an animal and had a remote trigger. We ultimately started building more elaborate rigs, such as the rig on the right, that could hold up to 42 cameras. And they're all synchronized. And this was, by the way, inspired by a lot of human work, because there's a lot of work done in Hollywood and all the zombie movies you've seen, where if you've seen people being eaten by zombies, they're typically everyday people who go to a Hollywood studio, they get themselves scanned, and they get given $50, and their body gets thrown into an animation, a movie. And I was interested in those techniques, started building these for humans. The systems are field portable, and they work in a wide range of settings. Here's a couple examples. This is a Beast Cam Array. It's a system that holds up to 42 cameras. You can put an animal such as in the bottom. It depends on the animal. It could be, some animals don't mind being turned over, some do, but it's a fairly non-invasive technique. This was funded by NSF, and it allowed us to go into the field and start photographing a lot of different small reptiles and amphibians pretty effectively. Here's another system that's even smaller than what we use today. We didn't use all these cameras. This holds 24 cameras. We call it Beast Cam Macro. It was developed by Michael Pereira and Trevor Mahan. One of the coolest parts of this project was seeing their growth, these students, and how they learned to develop this technology and the methods and then apply it later on. They're both engineers. And this system worked. I used sort of a downscale version of this because it turns out we don't need this many these cameras nowadays. Let's talk a little bit about shape. When you look at the slide, you see immediately all this variation in shape, from a hammerhead shark to a toad to a sea turtle to a leopard seal. But defining that shape in 3D is basically one of the main things that biologists do. And we argue about it, and there's huge debates about what shape is. There's no one way to do it. But what if we could have a tool that we could look at it more closely? So what we set out to do is really be wide-ranging with our initial approach and said, let's develop the methods and go after quite a wide range of animals to see if we could quantify the shape of these, and hopefully something good will come out of it. I do have colleagues in herpetology who were like, why are you working on rhinos? Are you okay? And I said, look, I mean, it's an opportunity. It's no different than measuring a lizard. It's just slightly larger. So we're talking about a southern white rhino, a great hammerhead shark, blue whale, and hummingbirds. Part one is Bakari. Bakari was this is a southern white rhino, an adult male. And one of the reasons we focused initially on rhinos, these are very critically endangered animals. We knew there was a tremendous amount of interest in their shape and their condition because climate change is impacting Africa in a very significant way. We know that these animals are probably losing weight due to seasonal changes and climate changes in rainfall patterns, but there's no good way to quantify the weight changes in something like a rhino. And so there was great interest in creating a 3D model of these animals so that people could study changes in body condition. How we did this is the animal was extremely well behaved. It ate hay, and we had a series of 20 cameras that surrounded the rhino. And I'd love to tell you it was anything more exotic than that, but it wasn't. It was very pleasant. And afterwards, I got to scratch Bakari's neck, which was fun. and that we're able to photograph it and then ultimately create a 3D mesh and a 3D final render with color. But it wasn't all that simple, because now you're asking the question like, okay, well, what about the under regions? What about the underside of the foot? How do we accurately reconstruct these scientifically? And this proved to be the biggest challenge. Turns out with a very dedicated zoo staff, this was much easier, because we would ask them, can you take pictures of all the different parts of the rhino and give us measurements and data? And they said, yes, we can, because Bakari is very well behaved. And so they would give us tons of measurements, and we had all, I didn't do these, they did this, and they took measurements of the underside and the underside of the foot. And through this process photogrammetry, turns out once you create the initial 3D mesh, you can take these individual photos and measurements and just literally insert them into the back end of the model and ultimately create this final 3D model, which can be used. This was the final result. And this animal, this is just a motion that I'm not going to argue is accurate. This was just added for fun, just as a typical motion, but it can be downloaded and used, and we were very happy with it. And I've been told by people who study rhinos, this has been a great tool for them to brainstorm about all kinds of research opportunities. But how will this ultimately be used? One of the next steps we're doing is to really estimate body condition from volume-mass relationships. One of the advantages of these 3D shapes is you get an accurate estimate of volume and surface area, which is notoriously hard to measure if you just have an animal in your hand. So we know the volume of this animal is 1,499,728 centimeters cubed. The surface area is 100,202 centimeters squared. Its mass at the time we photographed it was 2,225 kilograms, and we have length. What we aim to do is to actually use a machine learning approach, take this animal, create many new digital rhinos, and basically replicate surface area and volume, and then study that relationship, especially if we can get more rhinos and also get more data on mass, to then map out the relationship between volume and surface area and mass. And then we can come up with a really good relationship that we can use in the field. We're doing the same approach right now with elephants as well. Second part is something totally different, which is, what about animals like a shark, which I've worked with quite a bit. And I've been very interested in shark morphology, but measuring shark morphology when I've worked with sharks is very frustrating. You can go to a museum, you might have a specimen typically that are smaller sharks, or they're animals that are quite old or quite warped, so it's hard to really understand what a shark morphology looks like. When you work with, I have worked with sharks live in the field, but typically you're only given about 30 seconds to literally get next to the shark, measure everything, and then you leave, and then they let the shark go. Not very satisfying. This was a collaboration with the Angiari Foundation. Casey Sapp is a photographer, and the CG artist Jared Bott. But in the Bahamas, you have hammerhead sharks, which are great hammerhead sharks, which you can dive with. It's very safe. It's been done for a long time. The water is very clear. We understood a lot about these animals. They have very predictable movement patterns. We believe that we could do the same 3D system, and that's exactly what we were able to do. We set up a series of cameras. You can see them arranged around there. We have a person who feeds the animals, and I should say this is done every day there. This person, that's just his job. You feed the sharks, and tourists come. But sharks, it's just simply, these sharks are pretty mellow because they kind of do this, and it's kind of like when you go to the lunch cafe, you don't get too stressed out, you get your sandwich. And then that's us on the far side there. And that is a big, great hammerhead shark coming in there. We had one shark in particular that behaved very well for us. Her name was Nemesis. Nemesis was a tagged animal, about 10 feet long. What's fascinating is historically, great hammerhead sharks grew to 21 feet. So it blew my mind to work with this animal, to think, here's an animal that's 10 feet, which is a sexually mature shark, but just barely. These animals just get so big, but we just don't see these big animals anymore. That's just sad. From that 3D camera array, just like the rhino, we're able to reconstruct the shape of Nemesis. And again, because the Bimini Research Lab already had a lot of data on this shark, they had data on length and measurements and morphology, and I'll show you later, we were able to make sure this model was accurate. There's a photo of Nemesis, and here's the ultimate 3D rendering of Nemesis. I'll show you later what the full 3D model looks like. Again, available to download. We're now assembling, actually, using the same approach. I want to do more sharks. I'd like to do tiger sharks. We've done this with reef sharks. I'd like to do a lot of sharks to get a big shark morphology data set going. That would be a lot of fun. But it raises right away when you look at 3D model, you can see some features that are very interesting. For example, hammerhead sharks have this enormous sloped back. I've been on boats where people have actually caught sharks and they were tagging them and they had to be on the back, and the power of the axial muscles is incredible. They can just toss you off like nothing. But the slope, the changes, and the fin morphology. So this actually, I'm going to talk about later, offered us an opportunity to study the shape of these animals in a way that we never were able to before, because keep in mind, this is an adult hammerhead shark, which is great. Third example was a collaboration with Stanford University and Jeremy Goldberg and Paolo Segre and Johnson Martin as a CG artist. About they saw what we had done with the hammerhead shark, and they were like, could we do this with a blue whale? Again, one of the most elusive animals that we just don't have well represented in museums or really at all. And we'd like to get an accurate morphological model so we can study locomotion, so we can study movement. And I said, absolutely. In fact, you don't even need 4 cameras. What you need, which it turns out to have, is BBC came up with a series called The Hunt, where they had 1 camera filming a blue whale swimming all around. It was amazing footage. If you haven't gone back and seen it, I recommend you check it out. And we're actually able to reconstruct that. And this is published last year in Marine Mammal Science. But let me walk you through it. This is the blue whale on the bottom. And the bottom part of the photo, the gray part, is the 3D model. The top part actually shows the actual whale. So even with a single, you can either have many different cameras or videos surrounding an animal, or you can have one video that captures the animal moving in 3 dimensions, and essentially that amounts to the same. You actually can reconstruct the animal accurately from that. And that was exciting. They also had, by the way, drone footage. They were able to attach video cameras to the animal. You can see that in the lower right-hand corner. That shows you what a blue whale looks like, and they do these amazing dives. The thing that strikes you, so from this, we're able to reconstruct the animal, and it's available on Sketchfab, is down below. And then we can the blue whale offered new problems. Unlike a hammerhead shark, which we had morphology on, and Bakari, which was in a zoo, the blue whale just swam away, and you can't go out and measure it. And so how do you measure a blue whale? Turns out that people killed blue whales for hundreds of years and have tons of morphological data on them and had weight data, and we're able to go back and reconstruct the shape of it based on the size and the sex and the age and all that. You can see that basically the difference between the model and historical values is very low. So we had a lot of confidence. It was a very accurate model. One of the things that immediately came out of this that I really was excited about was blue whales are skinny. They're skinny. Big as a whale, we talk about that. And they're huge, right? 100 feet long. But this is a normal, when I say normal in terms of body condition, according to my colleagues, a pretty typical blue whale, which means that when you look at it from the side, this is the 3D model, it has this very large profile. But when you look at it from the top, I think you'd agree with me, especially that lower right-hand one gives a good sense for it. Really long, skinny animal. And this is actually one of the exciting parts about working with Jeremy and Paolo, as they said, this is great because now we can really begin to understand the evolution of size in whales. When whales get larger, one of the things they know is that that tail just keeps getting longer and longer and longer. But the actual shape of the tail was never really known to them. What Jeremy and Paolo really want to do ultimately is energetics. They want to understand the movements of these blue whales, how they expend energy. These are big open ocean travelers. They're incredibly fast swimmers, which is one of the reasons why they're so skinny. And this is actually a graph from one of their previous papers showing relationship between roll velocity when they roll. They do this to feed, for example, and roll angle. And so this is actual empirical data. What they actually want to do is create a computer simulation, put this model into the computer simulation, and see if these data values that they get in the field are accurate. And I'm really excited to see that. And I think this is one of the funnest parts of the project is I have a lot of scientific questions I'm interested in, but I also get a lot of pleasure about giving the tools to them and letting them have an exciting scientific questions. Because at the end of the day, I think that's really exciting, especially after COVID. I grew to appreciate this because I was so lonely hanging out with my kids. So in all, these are, we used this different varied approach to actually ultimately reconstruct a wide range of living animals, ranging from a southern right whale at the top, a rhino, a black-tipped shark, that's 1D, a harbor porpoise, number of sea turtles, reptiles. We do reptiles, Raina. See? Doing a snake was the hardest one, and they just do not want to become untangled. But one of the very valuable things I think we offer is a straight snake. It's very hard to get a straight snake. So when you scan a snake, you've got the 3D model, which was a snake from Cyprus. We actually offer digitally the ability to download it while straight, which turns out I think is extremely valuable. So among these 14 specimens we did this very wide survey. We were able to then look at parameters like surface area, volume. We had data on mass and morphology and asked some questions about shape. And the value of our collection, I believe, is that when you download them online, they're all properly scaled to length dimensions. And this includes the blue whale, which is really fun to put in there. I haven't done this yet, but here's the rhino, there's a black-tipped shark, there's a harbor porpoise, there's a flatback sea turtle, there's some geckos, there's some snakes. They're all scaled. So now we can ask some questions about how shape evolves. And this is one of the directions I'm very fascinated with. But before we do that, I think one question I get a lot is this accurate? Does this work across this very wide range of animals? And I think we have a lot of data to show that it does. Because we worked with animals that we knew certain physical dimensions, such as Bakari or a hammered shark or reptiles, we have live measurements on the x-axis, digital measurements on the y-axis for a wide range of morphological measurements, and the correspondence is extremely tight. I think it's about close to 0.99. We can start for these, just for that original slide I showed you, these 14 animals, this was published last year. We can start to ask, okay, well, what are some of the basic relationships between things like mass on the x-axis and surface area? We're talking about animals that have very different shapes. Do they all follow on the same line, or are they just radically different? And then on the bottom, we have volume and mass. And I was quite shocked when I looked at the data with my student Sana, and we saw that when you plot mass versus surface area, the isometric slope is really expected to be 0.66. The actual slope, and I should say I haven't done a phylogenetic correction yet. We haven't published this data yet. The actual slope is 0.64. This is a theme that I've seen throughout as we get more and more data. Every animal fits on a basic isometric line, which is interesting. Same thing below, mass and volume. Expected slope is 1. The actual slope is 1.01. There's a basic assumption out there among a lot of biologists that mass and volume scale 1 to 1. Well, that turns out they kind of do. But what about, as we've collected more animals, and my student Sana Seth, who's doing an honors thesis right now at UMass, we've been collecting a lot more. We actually have done a lot more 3D sharks that we haven't released yet. We've done a lot of turtles and dolphins. These are just aquatic animals. Let's focus on aquatic animals for a moment. And let's compare volume to surface area. Absolute critical parameters when we think about an animal swimming through the water. The isometric slope for this would be expected to be 1.5. And this compares, by the way, animals in quite shape, ranging all the way from blue whales at the very top down to the smallest sea turtle, which is a baby hawksbill sea turtle. The isometric slope is 1.5. The actual slope is 1.48, and the R-squared is 0.99. Basic relationships remain rock solid, steady. I still am blown away by this, and I don't want to publish this yet because I want to get more data. I find it very fascinating that these basic rule of volume and surface area kind of follow these basic rules. So I'm very curious to know how this turns out, what it means. Despite vast differences in body shape and ecology and life history, ranging from a lizard to whale, they all have the basic relationship, same basic relationship between mass, surface area, and volume, pending phylogenetic tests. And obviously, I'd like to get more data. I'd like to just keep adding to this. The fourth part is, and I think this may be of a special interest to folks here at the museum, because we developed the BSCAM system. It was a great prototype, and obviously it was really version 1.0. With Alejandro Rico Guevara, who's at the Burke Museum in Seattle, University of Washington. He's the curator of birds there. He was interested in, number one, could they develop their own system? Could we help them? And the answer was absolutely yes. We'll absolutely work with you. They were able to get a grant, purchase the cameras, and essentially take what we originally did, but improve it. And when I say improve it, make it feel portable, but make it lighter. They now need fewer cameras and actually better, higher quality. And I did this, I do want to give a lot of the credit to my PhD student, Josh Medina, who's a real whiz at this. And this, we called it PicoCam because it was originally designed to recreate the shapes of hummingbird beaks, which is what Alejandro was interested in. Now, you could say, well, why don't you just look at the beaks of a preserved specimen? And the answer is you can. But Alejandro knew that even upon preservation, the shape of that beak changes. And he was interested in the evolution of beaks and how they use them in fighting and conflict and a lot of fascinating questions. But this is the system. It has a lot fewer cameras. Here, it's field portable. We took it to South America. These are pictures from South America. There's Alejandro. He's holding a hummingbird. There's a picture right there. One of the things that we came out of this through using better cameras, better techniques, better lighting, essentially all the techniques of photography, is the usual rule of thumb of photography is 0.2 millimeters resolution. That has always been, if you read through over 20 years of work, kind of the standard. But we actually feel like we've exceeded that in the field too, which is great. And that was important because when you look at a hummingbird beak, such as, here's actually one on the left there, pretty freaky beak there. We're able to get very high levels of reconstruction. And actually you can sort of see that in this, our 3D reconstruction of the screw down below. And they actually have a trick where we put calcium powder on the beak, and the calcium powder allowed us, didn't hurt the birds. In fact, they got a little bit of calcium. And we're able to get all of what you see in the far left. These individual screw divots are far less than 0.2 millimeters, which is not quite what you get with a micro CT, but also not too far off. So the resolution was excellent. We're very happy about that. I think I'm going to do this at the end because I exited out before and it was a hiccup. So I'm going to show you a 3D model of a hummingbird captured in Columbia Wild Live later on. Motion. When we look at an animal, if we think about a specimen, I think the typical view is you have a specimen, it's captured, but could we reconstruct the movement animal? And obviously movement is highly variable. There's many different kinds of movements, but could we begin to understand what an animal does in the wild or maybe an aquarium and reconstruct what it would look like? I grew very interested in this, especially for sharks or whales, because when we think about migration patterns, obviously critical to understand movement, how sharks move, for example. So together with Vangelis Kalogerakis, Jianzhu, George Lauder from Harvard, we came up with a machine learning algorithm that is publicly available for anyone to do this. And it started to, I won't say it solved this problem, but it's a first step. And what this machine learning program does is you take, in part C, you take video of in this case, of a real shark. This was a cat shark swimming. You take that video, and this is just an animal aquarium swimming. I'll show you a video in a minute. And then you take that rig, and you also then add in your 3D model. This is a different shark. It's a black-tip shark that we scanned in Florida. And you essentially combine them. Now, obviously, a cat shark is not a black-tip shark, but proof of principle. It then takes the motion from the above, puts it in the model, and then you have your reconstruction. That's what it looks like. Let's see if I can get this to work. This is a video that kind of explains it. It's test case number 1. So that's a cat shark. That's a black-tip shark. So the motion from that has now been put into, this model, by the way, is not a very good model. It's one we did many years ago. There's another test case. We took a cat shark, put it into just a 3D model of a fish that he downloaded off some Sketchfab that was open access. There's another one. I think this was some 3D grouper that someone else scanned. So we wanted to do not only our own models, but other people's models. And we were pretty happy with it. And again, this is a sort of a two-dimensional, if you want to think about it, movement, but it worked out pretty well. So we never spent a minute creating that motion down below. That was just put in by the machine learning approach. I'm excited about this. Yeah, thank you. I'm very excited about that for kind of growing that, not just for aquatic animals, but also for terrestrial animals. Final part is color. So we're used to, there are already great techniques for quantifying color. We can think of light spectrometry. We can think of light, all kinds of ways. But what about applying this to a 3D method? And this is work really by Van Sasson, who's doing honors thesis, and Josh Medina. Again, this is his primary PhD work. And I've been very excited by what Josh has done. One of his main interests is the evolution of UV coloration in birds. We know birds can see in UV, at least some can, and they exhibit UV coloration that at least a human can't see. So he actually came up with a system just shown down below. These are preserved bird specimens. And here's what we see with a bird with our naked eye up above, and here's the UV patches that are exhibited on the upper right. That big white patch on their head is all UV reflectance. So it's a special kind of camera that could capture color in UV and also capture other kinds of RGB color that is more in the normal visual spectrum for something like a human. And then what he did is he actually had a turntable and create a 3D model of the bird and a 3D model of the color and combine them. This allows essentially Josh, Van, and I to reconstruct the color of this animal and then ultimately interpret it in terms of what a bird can see. So the workflow basically, and this has not been published yet, but essentially takes a 3D scan of the bird, a point cloud shown on the upper left-hand corner, creates a polygonal 3D model, and then creates a UV coordinate mapping of color, which is shown in the bottom left. So when you think about what a bird is not just color, they have these patches, right? And this is true not just in birds and other animals. And it's not just, you can't just think, oh, he's red, he's green, he's blue. They have colors, patches of each, and you have to understand how those different patches are. And so if you look at that bird on the left, the 1, 2, 3, 4, 5, 6, 7 represent different patches. And that actually shows, the one on the right next to it shows those patches that are taken from 3D and now put into 2D. And now we can quantify surface area, volume, and all the other basic parameters around them. But what we can create in the bottom right here is ultimately a full 3D color map of this animal. It's a little hard to see, but these are actually 4 different bird species here, and we've highlighted one patch. But what you can see here is we can quantify, for example, the percent total area of a certain patch. We can quantify how it fits in the RGB spectrum. And basically think of it as light speck, but all over the bird all at once. And more importantly, it's correlated to morphology. So the animal has a certain morphology. You can now map the color back onto the shape. So this is very exciting, and it allows us to understand, if you want to think about it simplistic, I've tried to put it here. These are 3 different bird species that are found in the tropics. And you can see some, obviously look incredibly colorful, some less so. And we know that in birds, there's tremendous variation in color. It's not always clear why there's variation in color, and there's differences between males and females. But this gives you a sense for it. It essentially creates a 3D color profile. So in the bottom right, you can see one bird, obviously extremely colorful, has a very large color profile. One on the far left, a much more narrow color profile. And you can break it up into UV coloration, which you'd see at the upper top-hand corner, or different levels of RGB spectrum coloration. So now what we're doing before we publish it is to really come up with good data metrics that we can calculate and compare bird 1 to bird 2, bird 3. And this is a technique, by the way, that's not can be used not just on birds, but on any specimen. It could be used on a rock, it could be used on a frog, it could be used on a fish, so forth. So to the coming conclusion, point of discussion that I would love to hear your feedback, and I do want to emphasize I'm just make points of discussion, which is, at least for certain kinds of animals, especially ones that are hard to collect in museums, this concept of the digital specimen has a lot of interest, at least for me personally. A concept that you say you have a 3D digital avatar that really is very accurate in terms of shape, color, and motion. I know the concept of digital specimen already has a meaning in the literature. I'm not trying to take away that meaning. I'm trying to argue that we can also think about these as potentially specimens that you can deposit potentially in digital museums that can be used for scientific research. And what we've done on the open access end is all of our models are open access. They can be used for nonprofit use. I'll open up the website in a minute, and some of them they get used a lot. We've only created 96 models, which is not very much, considering how much life is out there. But what we've been very excited about is the usage. Here's a profile over 3 years. I don't want to move from the mic, but as of the last week, we had 135, almost 134,000 downloads from 96 models. Pretty much shown in blue are views, shown in green are downloads, and you can see at the start of COVID, there was a dip, and then after COVID, people went, whoop, people are stuck in their homes. It's fascinating. But the usage is exciting, and as a basic projection, if we created 100 shark models, say we went around the world, captured 100 shark models using what we do, our projections, we have a lot of data to back this up, is that within about 5 years, we would have almost 500,000 downloads. And these downloads get used in a lot of different ways, by the way. They're used by kids, high school teachers, people who are geeks on sharks, scientists, artists. Actually, we still don't know what they get used for. So this is our pitch, basically, which is that I thought of Raina when I did this. Imagine all the incredible frog species in the world, the incredible diversity. Imagine all the great diversity of sharks, of which we captured only a very tiny fraction. And imagine if we could scale this up over many, many years. I think one of the things we can agree upon is a museum and educators, we want usage, we want education, we want collaboration and discussion. So the interest in this is exciting. I'll just end here, and then I'll open this up and I'll show some of the models, but I do want to thank funding. Again, so many people along the way. Here's our website if anyone wants to look at it. And then I'll just, let's see, do you think Ray and I can open this up? And let's see here, last time see here. Whoops. Yeah, okay, I think you said you have to do something.
Yes, let's see. I think we analyze this. Okay, oh, there it is, StreamYard. Okay, stop. Percent. Okay, did you want to show this one?
Yeah, why don't we start?
This one or this one?
Yeah, let's start with that one, and then we'll.
Okay. And then I think we have to move this over.
I see, perfect.
Okay.
And then I can go to the other one and pick the next one. That's fine.
I think so. Yeah.
Okay. Before I went in, I just want to show you. This was a 3D model of a hummingbird beak in the field. This was with the PicoCam. So it's again a live hummingbird that Alejandro is holding from below. He's the master hummingbird holder, and he's putting it up. And again, this was done with this new PicoCam system, which you can see here. It's a little hard to see in this light, but what I'm very happy about is the level of resolution. Let's see if I can zoom in here. You can see all the grooves and nooks and crannies which we were able to get. So very happy about that. And then the next one was, let's see if we can pull this open.
Click on, go here. Is that what you wanted?
Yeah, it's perfect. And then this is actually our website, which anyone can download models from. And I just thought I'd show you some of the fun 3D models. This was Bakari. This is the southern white rhino we did with the Perth Zoo. Bakari's still alive, but kind of gives you a sense for the model quality. I'm very happy with the rear end, actually. I feel like we worked very hard to do that, which is no easy feat, trust me. So yeah, I mean, I think this is exciting that I think this particular specimen, to give you a sense for it, has been downloaded 6,000 times, I think. We'd like to do more because the argument is that just what we notice is if you do one rhino, then you do two, people download the first one even more, just usage just keeps going up and up and up. And I guess I'll stop there. I think I've used up my time, but leave it with right. We'll leave it with Bakari sitting there staring at us. So thank you very much. I really appreciate it. Yep.
Firstly, thank you for the presentation. This is really cool.
Thank you.
The question I have is, what 3D software are you using to rig and animate and then upload to Sketchpad?
Yeah, we use Blender because I think being an open access software is such a huge plus. In the industry and like in Hollywood and entertainment industry, Maya is widely used, and Maya is a tremendously expensive software. But Blender has a nonprofit foundation behind it, and so Blender is our main tool. But we also use some. There's been some open access 3D reconstruction software. We use ColeMap, we use Meshroom, and we use other software like that. But yeah, and by the way, I think we'll probably be, yeah. I mean, I think that's when we actually work with partners, we actually spend a lot of time training students on how to use these things. So we spent many hours training people how to use Blender, which is, I think, for any of you students out there, is a fantastic piece of software to learn because it's, again, free. So that matters a lot. Did I answer your question?
Yes.
Okay, thank you. Good. Oh, sorry.
Sorry. Thank you. I have two questions. I have more, but I'll just
Sure.
keep it to for one, I'm assuming that you, especially for the rhino, in order to animate it, you went through a process of rebuilding the mesh, retopologizing it and such.
Right.
Are all of the models that you upload, has that been done to all of them?
Not all of them. Like a lot of I think, early on we didn't, like some of the frogs we did in the Philippines, and those were just kind of these raw mesh models that were just these huge models, and they weren't like, this is kind of a whole specimen, whereas those were like a frog sitting on a leaf. But we put them out there and they get used, and I think they're still valuable, but they're kind of like more of in the raw format. But yeah, you're right. This one went through that whole mesh retopologization process. Yeah.
And we actually have a use upcoming for the Harvard corpus that I'd love to download.
That's great.
Test it out for.
Awesome.
But another question is, what software do you use to do the photogrammetry?
Yeah, ColeMap and Meshroom are the two ones. I think it's such a strong, I don't mean to go on a rant here, but what happens is, there's commercial software like capturing reality, which is widely used in Hollywood and stuff, but very expensive, tyrannical almost. And then what happens is, I think Meshroom, I believe, or ColeMap was a PhD thesis from France. But of course, it's relentless how if you don't maintain that software, it just falls into, it doesn't get used. Like Blender has this nonprofit foundation behind it, whereas I don't think ColeMap and Meshroom do. So basically, it's a very hit or miss kind of thing. But it works, and again, I'm happy to train people on it. But yeah, so ColeMap, Meshroom, I'd say are the two big ones. But actually now, even in Blender, you don't need that 3D photogrammetry software anymore. You can put everything together in Blender by hand. I think photogrammetry, the mindset's always been take a bunch of photos, it creates the model. But now in Blender, you can just do it all by hand. It's really nice.
Like hand modeling based on.
No, I mean, you can align all the cameras yourself within Blender and you don't need that ColeMap and Meshroom really much anymore. The software, it's really the advances in the software that have changed everything. Yeah.
Two questions. First one, you talked about taking photos, 20 photos at once. Have you thought about taking continuous video to capture 2 minutes of motion, breathing, how the muscles move when a frog jumps? It seems like there's a lot that could be done there.
Yeah. Have you ever seen the movie Ghost in a Shell? Terrible movie. Sorry. But that's what they did. I mean, again, it's like crazy how Hollywood has this power. Like they took 60 frames a second video and just incredible firepower, and they reconstructed people in 3D. So I would love to do what they did, but the budgets for that are so high because typical video is the files are compressed, so you're losing image quality. But yeah, I think what you're talking about is like videogrammetry, which is, yeah, I think is out there and is usable. It's just, yeah, it's still, I feel, immature technology for people, humans like you or me. So yeah, we've tried video. It's just the quality is lower, and sort of like you're almost doing more work to make up for it, if that makes any sense. So I think I would say 10 years down the road, absolutely. I'd say that's going to evolve, and I think that would be a really exciting part, especially for the motion part. I agree with you. So.
You mentioned you've got something like 70 models. Do you have a plan to scale it up to hundreds and thousands?
Yeah, I think we have 96. I mean, we have more coming online. Like we're doing a bunch of sharks with Save Our Seas. Those will go online. We have essentially our business model. We're a cost center at UMass, so partners come to us, whether the scientists, nonprofit organizations. We work with a lot of nonprofits, and then they pay us. We create the content, and then just one of the stipulations is it goes online for free. But these projects do take a lot of time, because when you work with animals like this, yeah, you have to go through a lot of hoops, basically. So I would love to scale up to hundreds, right? More frogs would be cool. And yeah, I think it's really a question of investment. We need more funding to do more, but it's pretty hard to persuade. I don't envision SF ever wanting to fund anything like this, in my opinion, because it's just too, I don't know. I just, like, almost all our money's been raised by partners, but I just have a hard time believing NSF is going to fund anything like this, because it's not really hypothesis driven or anything like that. It's just like, let's create content. But who knows? I think with the color part, I could be wrong. So yeah, funding is always a struggle, but we've been able to essentially get a fair bit of money every year to do this work. Just keep going, I guess. Yeah.
You mentioned earlier that for the missing data in the northern regions, you had some zoo staff take photos for you.
Right.
Do you think there's any possibility that, I'm just thinking, in the field, like sometimes it's with like these hard to find or incredibly rare specimens that like take a lot of time and effort to just search out, and then you get there and kind of get skunked.
Oh, right.
Is there any way that the public, like maybe people that are in the area you're looking for, could take pictures and provide useful data so that like you can put it into these models and you can see, like, get a little more primary information as to like what you're looking at?
Yeah, I think it's a great point. I mean, I have a pretty close relationship with the Center for Data Science at UMass, which is a very well-funded center that does they've actually funded a lot of our work, and they do a tremendous amount of work in machine learning. One thing I've come to learn is the power, usually with my kids, I have 2 kids, I'm worried about ChatGPT and their essays, but the more positive side of machine learning is there's no doubt that's where it's all heading, because I am pretty confident in 20 years, I won't need to do any 3D modeling at all because of sourced photos and how machine learning works.
What kind of photos were the zoo people sending? Were they just like from their, did you give them cameras, or were they just from their phones?
Oh, they had their own cameras. I mean, essentially they do vet checks where they once a month go into the animal cage and kind of look at the animal and take photos and do measurements. And I just said, if you're going to do a vet check, could you take some photos and share them with us? And they said, fine. But yeah, I think you're right. I think it's a fascinating question to ask. Could you fill in an animal, the gaps? And it's fascinating because if you have a 3D model, let's say it's missing a toe, you can, if you take an image, you can just slap it in there and it's quite accurate. Yeah, I think that's great. I think the other point about you guys being a museum is that if you scan, say, a dead specimen, and then you have a color of a live specimen, and we've worked on this with fish, you can just slap that color on for the fish, and you get a very accurate 3D model. So like you scan a dead sea bass, a picture of a sea bass, you can, I have an example of that, actually, I can show you later. So great questions. I totally agree with that. Yeah.
Like in that same grain of what you just mentioned, what do you think this is a strange question, but what do you think the potential is of taking a scanned dead specimen and adding on color from a live illustration? Yeah,
we've done that. I can show you an example of that. We did that with fish. We've actually done that. We've actually done it's sort of fusion, like we've CT scanned like spiders, and then we've taken images photogrammetry scan and fused the two 3D models. So it's kind of a Frankensteinian monster because they are two different animals, but it works. It's actually really exciting. Yeah.
I asked the question because we have here specimens that were collected on expedition and in the 1900s.
Sure.
Early 1900s.
Like they're preserved specimens.
They're preserved. We have
Sure.
them in the collection, but somebody did like live
Photograph.
animal
Oh, yeah.
watercolor paintings in the field.
Oh, really?
And sent an illustrator to the field, and so there's like there's beautiful watercolor images of the fish, but then the fish is dead in the collection.
So yeah, absolutely. That I believe that can definitely be done because essentially, if you think about it, if you have a 3D model and you have a 2D image, what you do is it's a wrapping process, and it's just a question of establishing your coordinates. That's kind of what that's done in the software because the software creates a 3D color map and a 3D shape map, and then fuses them around it. So you can take photos and wrap them. Yeah, so I think that would be, I'd love to talk to you more about that. That would are these frogs?
The fish.
Yeah, that sounds really cool. Yeah, I'd love to have, fish would actually be much more straightforward. I can show you an example of a pumpkin seed sunfish we did this with. We had a picture of a pumpkin seed sunfish and we had a preserved specimen. There were different animals and we just wrapped around it. Well, maybe I'll try and pull it up. I don't know, see if you're, let's see there. Oh. I don't know if I'm doing this right. I'll just,
I'll try to help you.
okay, sure. Okay, yeah. More questions?
Yeah, I'm curious if you, in a similar vein, do you have animated specimens where you have like internal morphology, and like look at that motion internally and
Yeah. It's funny because one time I talked with folks from Purdue and they run like a giant commercial beef, like they do a lot of the farm beef stuff. And they're like, yeah, we love the 3D model of a cow and all the beef parts. Like, I think they're already doing, essentially they're like, we can get you scanned all the cows you want. And it was just totally interesting to talk to them. But yeah, absolutely. I think what we've been doing is more focusing on creating the external model and then providing as a tool. But from what I understand of the many emails I get is there are people doing just that right now. Like we're not doing it, but they are. Oh, this is great. Thanks.
Okay, I'll let you find it.
So yeah, I think that would be exciting. It would be actually really cool to CT scan an animal and then to get a photogrammetry scan and fuse the two and have like internal organs. That was done, yeah, I think it's a Natural History Museum, did it with a mummy. They did a 3D photogrammetry scan of the outside, and then they did a CT scan of the inside, and they fused them. So the technology is there. It's just these processes are sort of not well established in our community, but yeah. So sunfish. Let's see here. These are some of our models, but just for fun kicks. There's really fun stories behind all of them. But here's the, to go to your point, there's a bluegill sunfish. And that was from a museum specimen. And then what we did was, one of my favorites is the jumping spider, which is a fun project. Can we, I think,
Bluegill's right there on the left.
yeah, I think that's it. We did. I think this might, there's actually another bluegill. This might not be the one, but I think it's a similar idea. So, and we did it was fun. We did a little aquatic background, which was just for fun. This was meant to represent like the James River. I think there might be a different one actually. Let me see here. But yeah, so the bottom line is that was done in that way. But let me see. I'm pretty sure there's a different, all the sea lions. I'm pretty sure there's another sunfish in there. I swear. Largemouth bass. Some of these, by the way, I do want to emphasize, some are better than others because we've grown along the way. There's a straight snake, rhino.
Duncan, I just had one
one question.
Yeah, sure. Oh, here it is. Sorry. Go ahead.
Oh, that. The kind of like ballpark of how much some of these rigs are.
The 3D rigs. Whoops. It depends a little bit. I guess if I were in your shoes, I would do a little bit more what Alejandro did and focus on the PicoCam type system. Because I think it's a big step up from essentially what we're doing today. Although what we're doing today is fine. I mean, I think he spent, $30,000 or something. When you look at all the camera technology and all the goodies together, something like that. But I know they're getting tons of data, so it's a good investment. I mean, they're just going crazy over there, scanning. Once you get it, the beauty is there's really no additional costs because the software is free, which is nice. So yeah, something like that, I'd say, Raina. Go ahead.
So, sorry, another question. I work in exhibits, make models, and one of our big barriers to making accurate models is the cost. And so I'm also curious in terms of costs, like how much, do you have a way of, do you record or calculate like how much labor goes into the creation of one of these models? And obviously they're going to go, anyway.
Yeah, you're right, like human cost. I mean, it's a good question. I think one thing I was talking about with Raina was that what I like about this, I view it as a training thing, especially since COVID. I've become keenly aware how students have been a deficit of learning. And so we actually have a huge lab where we do a lot of training, not just on photogrammetry, but on 3D modeling, because a lot of these techniques are widely used in dentistry and medicine and engineering. And actually they're used everywhere, honestly. They're very powerful technology. So I view it as like my personal thing. I want to train students in skills that they can use. But typically I found that, undergrads can learn this really easily and they gravitate towards it. So we usually have an army of undergrads that's able to do almost everything from the beginning to the end. So for me, selfishly, it's free because they're all independent studies. Now, if you're hiring staff, it's a good question. I think there is, the biggest thing I would say, what we worked with Alejandro was, I think it's an investment of time and energy. So it's skill development essentially. But once you have those skills, you can produce so much data. So I do think it's going to take some time to get a team up to speed and build protocols. But once you have it, it's great. So I can't really answer that question effectively, but I'd say it's nothing that's too extremely unusual. I mean, if students want to learn, I mean, come have them talk to us. And we're thinking about offering workshops in this. I mean, that's some virtual workshops and things like that. So. Yep.
One more question. Have you worked with elephants at all or done scanned any elephants?
Yeah, we're doing some work with elephants in Africa, and that's been a lot of fun. So it's the same interest body condition because same thing with rhinos, climate change is impacting water distribution, especially in northern Africa, northern Kenya and Tanzania. And so like when I was there, it was interesting. Like the elephants, it was so dry, and animals were dropping like flies because it was great for the predators, not so great for the herbivores. But I think there's just been interest, like how do you quantify how much an elephant gains and loses weight seasonally? And I think so. I view that as a conservation thing. Yeah, and I'd like to do more elephants. That would be cool. But first off, we got to finish the one that we're doing, I guess.
Do you have any idea of when that model might be available, if at all?
It's done. It's just got to meet all the approvals, and probably it'll be out in the next couple months, hopefully. Yeah, really excited about it. His name is Sarara, which was actually an elephant that we filmed and then was later killed. So Sarara was speared was initially speared and recovered, and then later on was killed by locals. So very sad, but.
Okay, one last question.
So the photogrammetry I've seen, either you take a lot of photos of something at the same moment, or you have something static, and you walk around and take photos from different angles, taking your time.
Sure.
So the blue whale jumped out at me. The shape is changing, but you use a series of videos So how did you,
Oh, the shape of the animal.
how did you reconstruct the shape when the shape was different each breed?
That's interesting. Yeah, I mean, I guess, I mean, you're right. Certain animals definitely would change shape. I always think of cats, or we've worked with sea lions when they swim. But when blue whales swim, I think they're just like sharks. Their bodies become fueled, like hydrostatic, much more stiff. That's how they swim fast. Their bodies, like sharks, when they swim, their bodies become very stiff. And so their body, actually, I don't think the body really changes shape much, especially, that's my understanding. But you're right.
The tail moving back.
Yeah. Well, yeah, it's a good question. I think there are a lot of averaging algorithms you use in the blender and other that kind of get you the shape. They kind of look for error corrections. But you're right. I think you've hit on a weak point, which is what do you do with an animal like, well, let's take a harbor seal, which are these really floppy, like giant balls of blubber, and make them more, Yeah, that's a good question, and they also just gain weight seasonally. So I don't know. I don't have a good answer to that. That was our best reconstruction at the point in time. But your argument's a good one. That what would you do if you had an animal that was like really like jellyish? And I don't have a great answer for that. So, but, yeah.
Sounds like your reconstruction wasn't automated, it was labor intensive.
Yeah, because you're doing landmarks. I mean, again, it's all spelled out in our paper, but I didn't want to go into gory detail today. But essentially, it's kind of like 3D geomorphometrics. I mean, I would love to spend more time going into it, but essentially, you know certain marks of the animal, so you're sort of marking it in 3D space, and you're tracking it through different frames and making sure that those all line up. I mean, that's kind of it in a nutshell. Maybe next time, I'm giving this talk at a number of different places. Maybe I'll add in some more. By the way, you guys, this is all brand new for you guys. I've totally created this whole talk new because I was excited by this, and I thought you guys would have some interesting thoughts and comments, but maybe the next one I'll give a lot more details on the methods. That'd be great. But yeah, I think that I'd say blue whales are quite, the only difference was there, I'll say, is feeding. Jeremy says I couldn't release this, but he didn't say I couldn't show it. But, maybe it's hidden. Yeah, sorry, it's hidden. But this is a blue whale when it's just swimming, but we also reconstruct blue whale when it's feeding. It's just crazy. But that was a far more sophisticated project and was just capturing its fullest point of inflation. So I'd have to log in and show you the behind the scenes ones. Yeah.
All right, thank you so much, Duncan.
You're welcome. Thank you. Do you want me to?
Get out of here.
Yeah, do you want me?
I can do it.
Just delete the quick.
Oh yeah, I can do that too.
Sorry.
I just wanted to.
Hi, Duncan.
I'm Lauren.
Lauren.
Nice to meet
Oh,
you.
yeah, great.
Yeah, I was hoping
Close
to
by.
meet with you later this afternoon.
That's great. I only have to leave probably about